This is a student project I did when I was an undergraduate last year. I graduated summer just passed. Yeah, unfortunately, Paul couldn't make it today. So he asked me yesterday if I could give a talk for today. So I've had about 4 or 5 hours to prepare for this. So please bear with me. I have this really interesting project where we looked at Carex ornithopoda, bird's foot sedge, and we looked at the genetic diversity and population connectivity. And one of the things we sort of investigated with this was the limestone pavement connectivity across the UK. So as I mentioned, this was a student project done by myself. I was supervised by the lovely Jen and Paul, and just a little thanks to our extensive sampling team that helped us cover this. And when looking at sort of population connectivity and variation, the Lawton report in 2010 comes to mind with this quote, which says there's compelling evidence that England's collection of wildlife sites are generally too small and too isolated, leading to declines in many of England's characteristic species. This was said by Professor John Lawton himself, and in context of our sort of population connectivity and variation, essentially what is meant by this is when discussing collection of wildlife, we need more, bigger and joined up groups. So when we put this into the context of genetic diversity and connectivity, essentially what's meant by this is we're seeing a loss of genetic diversity, and with that we're seeing reduced resilience to environmental change, and effectively plants' ability to respond to environmental change. And what we need are larger populations that are more likely to have higher levels of diversity, and with that we'll get more connected populations and also more likely to be genetically diverse. So coming back to the sort of Lawton quote that I mentioned before, we get this ideal sort of groups of or populations of plants which are large, genetically diverse and sort of interconnected populations. We can see genetic information coming between groups where genetic information is getting between all of them and they're all sharing genetic information. But in this sort of Reedian reality, anyone who's unsure of Steve Reed, he's our current environmental minister. I didn't know that. So and what we are seeing is these sort of, I don't know if this green one is clearly smaller, but we're seeing these smaller genetically disproportionate groups that aren't exchanging as much genetic information, and we're effectively getting isolated populations. And sort of the things that are causing this and the factors affecting our genetic diversity and connectivity are things like reproductive barriers or geographic barriers. These can be natural things like rivers, mountains, those sort of things, or they can be artificial, like main roads and infrastructure development. And that's causing this habitat fragmentation, where in the case of our limestone pavement, they're sort of separated from across the country. You know, we've got big clumps together or in Lancashire, the Dales, but also smaller groups in sort of reaches of northern Scotland, that sort of thing. And it sort of encourages discussion on dispersal mechanisms of plants. So how is pollen and seeds dispersing? How how far are they dispersing? Can they get to these other limestone pavement and calcareous grassland sites? And these natural, for example, these natural reproductive barriers, they can sort of be enhanced by climate change. It all comes back to this root problem of climate change. It develops these reproductive barriers, and plants and taxa sort of migrating to accommodate their newer niches as climate change develops. And it can force plants into these fragmented areas where they become isolated and they can't exchange genetic information and such. And in the case of our limestone landscapes, these environments are very rich botanically in biodiversity, with a lot of nationally rare and scarce species, as we've seen a lot in today's discussion. So they have a lot of they have the high nature value, and it's important to discuss that these environments are at most risk from climate change, and that's highly emphasised in the UK. So to study this population connectivity and this limestone pavement calcareous grassland connectivity, we're looking at Carex ornithopoda, bird's-foot sedge. It's restricted primarily to northern England on cold, well-drained upland limestone outcropping, so along like limestone pavement ridges where it meets calcareous grassland, that the neutral grassland, that sort of thing. And as I mentioned, these limestone pavements are most at risk from climate change, and I've pulled this figure from the paper that is currently in press. The Carex ornithopoda distribution is on the western edge of what we're seeing in its global distribution. So when we start seeing climate change develop and the UK becomes like a less temperate, more warm place, we're going to see populations of Carex ornithopoda move and migrate to accommodate their new niches, which can be restricted to potentially the more northern ends of Scotland, where those limestone pavements are incredibly fragmented. So that brings us on to our research aims. Okay, so we had two main questions. How diverse are the UK populations of Carex ornithopoda? And how genetically connected are they? We're essentially using genetic connectivity and genetic variation as a sort of proxy to measure how fragmented are these limestone pavement regions and like calcareous grasslands. We also have a comparison with Estonian population that's a more normal central distribution of the sedge. So collecting our birds-foot sedge, we use the BSBI database. Across 8 sites sampled from England, we had 113 individuals and 50 samples from 5 populations in Estonia. Figure on the screen is also another one I've pulled from the paper and press, just highlights the spread of populations. Each blue dot represents a site we visited to collect leaf tissue from. So we have a big cluster in the Dales, one in Whitbarrow in the Lakes, up as north as Coombs Woods in just south of Penrith, I believe, and further south in the UK in the Derbyshire Dales, I believe. So how are we measuring genetic diversity and connectivity? We're using microsatellites, short sequence repeats. A lot of I'm not sure if a lot of us in the room are geneticists or as enthusiastic about genetics as I am. Essentially, they're conserved and non-coding regions. Their sort of variation is coming from base mutation rates. They're really good for studying base level genetic variation, as they're not under any selection pressures. And from that, you can identify variation. It's sort of like how they do it on the Jeremy Kyle show, actually. So if that's any stimulation for how that works for you. So this is the lab work. I won't bore you with all of this stuff, but if that's those sort of complexities of like the DNA extractions and the fragment analysis interest you, we can talk about afterwards, and that sort of stuff. So that allowed us to generate quite a lot of data, a lot of figures, a lot of calculations and that sort of stuff. But I pulled out a few main findings to discuss with our genetic diversity. So the figure on the screen is looking at observed and expected heterozygosity according to the Hardy-Weinberg equilibrium, which essentially looks at the extent of heterogeneity and mixing of groups within a community. So I'm comparing the English and Estonian populations And the sort of main thing I want you to gather from this is our observed heterozygosity, which is our yellow bars. There's a bigger value of heterozygosity compared to our English groups, which is essentially meaning they're mixing more genetic information compared to the English populations. And that can be highlighted a little bit further in few figures I've made here, which I've also pulled from the paper. We've got a principal component analysis, a spatial one, where I've taken the genetic data, geographic data, and analysed it and used colour. So what the colour represents here, each dot represents the populations we sampled. The colour represents genetic distinctiveness, so how similar they are to each other. So if we look at this little zoom in here in the sort of Yorkshire Dale region, we've got these bright green colours here. The sort of more closely related they are to each other means they're more similar in colour. So we've got like this big patch of green in the north, okay? And we're seeing that those populations in the Dales and a little bit further south, they're mixing genetic information with each other. The pollen is dispersing, the seeds are dispersing, or however it works. Excuse me. But what you can notice immediately is if you look towards the lakes, the Whitbarrow, it's a completely different colour. It means it's genetically distinct from those groups. So something is preventing that genetic information from getting in between them. Something is active as a reproductive barrier. And we're seeing that also, what's the word, emphasised when we look towards Crestbrook Dale, where it's a brighter blue colour. So that's also showing genetic distinctiveness. So they're not, genetic information is moving in and around the Dale. It's getting a bit towards the north of Cumbria, but it's not getting towards the lakes and Crestbrook Dale. There's something active as a barrier. And when we compare that with an SPCA of our Estonian groups, there's not a lot of variation in colour. You get a little bit of a brighter blue, you get a little bit of a darker blue. So genetic information is moving a lot more freely within Estonia than it is in the UK. So it's sort of emphasising the habitat fragmentation. Genetic information and plant material isn't moving as freely between limestone pavements and these calcareous grasslands as they are in England compared to Estonia. So what we're seeing, if we come back to the sort of Lawtonian ideal I mentioned before, this is the sort of reality we're seeing. We're seeing variable sizes in populations, and they've got variable genetic diversity. And it's showing mixed interconnectivity. Genetic information is moving freely between some, but not between others. So does things acting as reproductive barriers or just general barriers for these limestone pavements and calcareous grasslands? They're diverse, but not always connected. So, for example, Coombs Wood was our northernmost site near Penrith that can actually demonstrate the long distance dispersal capabilities. It was separated from that central Yorkshire Dale group where the source of the River Reading is, and as you move north along the River Reading, you can find a few samples of Carex ornithopoda sitting there genetically related to them. So we're sort of assuming that dispersal, maybe by seed, maybe by and germination, or it can be vegetative, as we're seeing with Coombs Wood. Yeah. So the sort of main discussion points we're looking at is genetic pattern ancestral or as a product of current land use. And essentially what that means is, have those populations always been there, or is current land use, like sort of the habitat fragmentation, producing the connectivity and variation we're seeing today? And one thing we could look at is Carex digitata, a very closely related sedge to Carex ornithopoda, is sort of a lowland edition of the highland Carex ornithopoda, and it shows similar patterns. But the thing is, the BSBI maps are quite unchanged. So we think the sort of succession of this sedge can be upwards of 7 generations may have existed, and that could be as a result of sort of pre-industrialization farming that where that interconnectivity is sort of stemmed from there. Our implications for conservation, we sort of discuss that the species recruits from seed rather than vegetative growth primarily. So to promote the interconnectivity and exchange of genetic information, overgrazing is sort of showing a threat to this species in particular. So when grazers eat the seed heads, it's preventing seeds from transferring between sites and that sort of thing. So we could aid the interconnection of the species through reduced grazing from sheep, ideally during the flowering season. That sort of thing. That's how we can promote the conservation of this rare sedge species. So further work could be looking at the longevity of Carex ornithopoda. Do we see the same pattern of variation on these other karst landscape specialist species? And how does the variation pattern of Carex digitata compared to Carex ornithopoda? And yeah, that would be the further work for us. Yeah, so this was, like I mentioned before, this is for a project that that I did for my undergraduate. We revised, is currently in the press. I actually have my email and LinkedIn screenshot up on the board, as I'm a master's student coming out into the working world within the next 3 or 4 months. So I'll be looking for the working world. This you could call this a sort of self-advertisement. Yeah. Any questions?